Dear students, welcome to E-Pathshala. I am Dr. Sunil Kumar Onteru, a senior scientist in Animal Biochemistry Division at National Dairy Research Institute, Karnal. Today, I am going to teach you about ion exchange chromatography under the paper Biochemistry. Our main objectives of this lecture are to understand the basic concept and principle of ion exchange chromatography, to understand the type of ion exchangers and also the choice of ion exchanger, and to know about the eluent pH and procedure of ion exchange chromatography, and finally we try to understand the applications of ion exchange chromatography. The principle of ion exchange chromatography is the separation of analytes based on the attraction between oppositely charged stationary phase particles and the analyte particles. The stationary phase in case of ion exchange chromatography is called as ion exchangers. Why? Because it helps in the exchanging of ions on the surface of the stationary phase. There are two types of ion exchangers. One is called as anion exchanger, which helps in the exchanging of anions, and another one is called as cation exchanger, which helps in the exchanging of cations. If you see this picture here, the anion exchanger, in this anion exchanger on the surface there is a positive charge. Because it is anion exchanger, it has to attract anions which are negatively charged. That is why on the surface of anion exchanger there are positively charged groups. So, these positively charged groups can attract negatively charged ions on the surface. So, whichever has higher negative charge, that can exchange the existing negative charge molecules present on the surface or attracted on the surface to the anion exchanger. The similar principle works for the cation exchanger. In this case, the surface of the cation exchanger has functional groups that has negative charge. So, these surface negative charge can attract the positively charged molecules on the surface. So, whenever there are two different kinds of positive charged molecules encounter on the surface, the higher strength positively charged molecule can replace the lower strength positively charged molecules on the surface. That is why there is an exchange of ion from one cation to another cation on the surface of the cation exchanger. So, this is the major principle of ion exchange chromatography. Let us see what are the differences between anion and cation exchangers. The major difference, as you have just seen in the previous slide, that the anion exchangers have positively charged groups on stationary phase and cation exchangers have negatively charged groups on the stationary phase. The another difference is the anion exchanger attract the negatively charged ions on its surface and the cation exchanger attracts positively charged ions on its surface. The third difference between anion exchanger and cation exchanger is the anion exchanger is called as basic ion exchanger. Why? Because the charges present on the anion exchanger is the positively charged. These positively charges are formed by the association of protons with the basic groups. That is why the anion exchanger is also called as basic ion exchanger. On the contrary, The cation exchanger is called as acidic ion exchanger because the negative charges present on the surface of acidic ion exchanger or cationic exchanger are mainly formed due to the ionization of acidic groups. We can see the examples of anion exchanger and cation exchanger. Here we can see examples for anion exchanger and cation exchanger. DEAE cellulose is considered as one of the best examples for anion exchanger. As we just now discussed that anion exchanger, there is a positive charge on its surface. Here we can see the positive charge is at the level of NH plus. So, the proton which is attached to the nitrogen in the ammonium ionic form here, that is contributing the positive charge. That is why this exchanger is also called as basic ion exchanger. On the contrary, in case of cation exchanger, the best example for the cation exchanger is CM cellulose. In this, the negative charge is because of the ionization of the carboxylic group. So, when the COOH is broken down into H plus and COO minus, this COO minus is contributing the negative charge on the cation exchanger. So far, we have discussed about the kinds of exchangers as anion exchanger and cation exchanger based upon the charges present on its surface. Let us discuss the classification of ion exchangers on the basis of the strength of ionization. The ion exchangers on the basis of strength of ionization are classified as strong exchangers and weak exchangers. The strong exchangers means ion exchangers with functional ionic groups that are totally ionized at all working pH values. That means, if any experiment has to be conducted at any state of pH situations, during that wide range of pH situations, a strong exchanger should be used because it can maintain the functional ionic groups on its surface on a wide range of pH values. The best examples for this are the sulfonate ions and quaternary ammonium ions on the strong exchangers, which can maintain the ionic state in a wide range of pH values. Weak exchangers are those ion exchangers on which the functional ionic groups that are ionized over only a narrow pH ranges. The examples for this is carboxylate ions and diethyl ammonium ions, which can maintain their ionic state in only a narrow pH ranges. The choice of exchanger mainly depends upon the stability of the test analytes. If the test analytes, especially proteins, are stable at a narrow pH range, then an exchanger that can operate in the narrow pH range can be selected. If a protein is stable below its isoelectric point, then we need to use cation exchanger. Why? The main reason is proteins below its isoelectric point, they will have net positive charge. When they have net positive charge, we have to use cationic exchanger. But if the protein is stable above its isoelectric point, that means at that stage these proteins will have negative charge on its surface. In this case, we need to use anionic exchangers, especially like CM cellulose kind of exchangers. If a protein is stable over a wide range of pH values, then we can use either cationic exchanger or anionic exchangers. Similarly, just now we have discussed about what kind of exchanger we have to choose, either anionic exchanger or cationic exchanger. Let us discuss about whether what kind of exchanger, whether it is a strong exchanger or weak exchanger do we need to choose. Strong exchanges need to be used for the separation of weak electrolytes, as weak electrolytes need very low or high pH for ionization. In general, the weak electrolytes, they need very either low pH values or high pH values to be in the ionization. I am taking pause. Okay. Generally, the weak electrolytes, they need either low pH values or high pH values to maintain their electrolyte state. So, in that case, we need to use strong exchanges, because strong exchanges can maintain their functional groups on its surface in a wide range of pH values. That is why we need to use strong exchanges when we are separating the weak electrolytes. But strong electrolytes, we can use weak exchangers for two important reasons. The first reason is the weak exchangers, they will maintain their ionic state in a narrow range of pH values. Generally, at this narrow range of pH values will be close to the physiological pH. So, there is a probability that there is a reduced tendency of protein denaturation at this pH values. The second reason for using weak exchangers for separation of the strong electrolytes is the inability to bind weakly charged impurities and promoting the elution of impurities, especially when you use weak exchangers which work only at a narrow range of pH values. At that pH values, some of the weakly charged impurities, so they cannot bind to the weak exchanger, that is why they will be eluted out and only the required molecules are bound to the exchanger. Dear students, so far we understood about the principle of ion exchange chromatography, types of ion exchangers and how to choose an ion exchanger. Let us discuss about the pH of eluent that is used in ion exchange chromatography, the procedure of ion exchange chromatography and applications of ion exchange chromatography. Let us discuss about eluent pH. The pH is a very important thing in case of ion exchange chromatography. The buffers that are used in ion exchange chromatography should have either one pH unit above or below the isoelectric point of the analytes. This is very important point. The reason is the pH either one unit below or above the isoelectric point will help to maintain the ionic state of the analytes. That is why we need to choose the eluent pH or mobile phase pH at least one unit above or below the isoelectric point. Anionic buffers. Anionic buffers are used for the cation exchange chromatography and cationic buffers are used for the anion exchange chromatography. The examples for anionic buffers are the tris, pyridine and alkymine. But the examples for the cationic buffer are acetate, barbiturate and phosphate. The initial buffer pH and ionic strength should allow the binding of analyte to the exchanger. So, whenever we are starting our experiment, the pH of the sample should be in such a way that that will promote the binding of analytes to the exchanger. Similarly, the ionic strength is also important in this particular sample buffer. Why? The specific ionic strength only promotes the binding of analytes to the exchanger. There should not be low ionic strength or there should not be high ionic strength. There should be an optimum ionic strength. Especially the lowest ionic strength buffer should be used initially to allow the minimum binding of the contaminants. But in case of gradient elution, initial conditions should be chosen that the exchanger binds all the test analytes at the top of the column. So, the buffer pH should be in such a way that will promote the binding of all the test analytes to the exchanger at the top of the column. Elution, gradient elution is common in case of ion exchange chromatography than the isocratic elution. For anionic exchanger, the pH gradient has to be decreased and ionic strength has to be increased. When the pH gradient is decreased, that means the H plus ion concentrations will be increased, and the proteins, when there is a decrease in pH, there will be more of cationic state rather than anionic state. So, basically here the anionic state of the protein will be decreased so that they can be easily replaced or displaced with another anions present in the sample buffer, which are generally chloride ions. In case of cationic exchanger, the pH gradient and ionic strength both have to be increased. What happens here is when pH gradient is increased, the cationic form of proteins will be decreased. That means they will lose their cationic state and they will enter into the neutral state or anionic state, so that on cation exchanger the proteins can be easily replaced with another positively charged ions, generally in this case sodium ions. So, let us see the procedure of ion exchange chromatography in the next slide. There are majorly 4 steps for the ion exchange chromatography when we perform the experiment. The first thing is equilibration, and next step is sample application and wash, and third step is elution, and final step is regeneration. Let us discuss these steps one by one in coming slides. Here we will discuss about the first step, that is equilibration of the ion exchanger. What does it mean? The charges on stationary phase need to be equilibrated with exchangeable counter ions available in the buffer. For example, the chloride ions for anion exchanger. Chloride ions are negatively charged ones, and anion exchanger contains positively charged on its surface. You can appreciate this on the figure present in this particular slide. Here you can see on the surface of anion exchanger there are positively charged functional groups, and the chloride ions present in the sample buffer, they will be attracted to the positively charged ions. There will be an equilibration state between the positively charged and negatively charged ions. So, by this step, all the charges present on the ion exchange stationary phase, there will be equilibrated with the negatively charged ions present in the sample buffer. So, this is very important step. Why? Because these negatively charged ions will be replaced with the negatively charged our analytes in the anion exchange chromatography. The goal of this step is to allow the binding of required molecules to the stationary phase and eluting the unrequired molecules. The sample buffer should have the pH and ionic strength similar to equilibration buffer to promote the binding of required molecules to the surface and unrequired molecules to be eluted out. In this picture, we can see that all positively charged molecules are eluted out because there will be repulsion between positively charged ion exchange surface and positively charged molecules. That is why they will not bind on the surface of stationary phase, that is why they will be eluted out. On the other side, the negatively charged molecules present in the analyte, they replace the chloride ions which are already negatively charged from the surface, so that the negatively charged molecules will attach to the surface of the stationary phase and chloride ions will be eluted out. So, by this the required negatively charged molecules are attached on the surface and positively charged molecules and exchangeable chloride ions will be eluted out of the column. For this purposes, the sample buffer should have pH and ionic strength similar to equilibration buffer, so that the chloride ion concentration will be maintained as if the similar to the equilibration buffer. The required molecules exchange the similar counterions already adsorbed on the column. That is what we just discussed, that the negatively charged molecules present in the sample, they replace the negatively charged chloride ions on the surface in anion exchanger. In case of cationic exchanger experiment, the negatively charged analytes and also the replaceable sodium ions, they will be eluted out, and the positively charged proteins that are attached or adsorbed on the surface of ion exchanger which has negative charges on its surface. The third step in ion exchange chromatography is washing and elution. After sample application, we have to wash the column to remove all the impurities. This can be done by using either sample buffer or the equilibration buffer. So, once we have done washing, then we can use elution buffer. Elution buffer should have sufficient pH and ionic strength to obtain the required molecules from the column. As we just discussed, in case of anionic exchanger, the pH gradient has to be decreased. Similarly, the ionic strength has to be increased. But in case of cationic exchanger, the pH gradient of the elution buffer should be increased and ionic strength should also be increased. In both the situations, the ionic strength should be increased. So, when we do washing step, what will happen in case of chromatogram? You can observe in this picture the first peak, which is a very broad black color peak, that indicates all the impurities that are not attached to the column and that are eluted first. At that situation, you can see the pink color line or the dark pink color line that represents the ionic strength of the sample buffer or equilibration buffer. Whatever may be the concentration or ionic strength of the equilibration buffer, we have to maintain similar ionic strength during washing step. That is why the ionic strength line is flat at initial stages where there is a black peak present. Later on, we have to increase the ionic strength. As we increase the ionic strength based upon number of charges present on the proteins which are already attached to the surface, they will be eluted out. For example, the pink color ball which is present, or one ball which is attached, or one protein molecule which is represented in the pink color, it has only one negative charge, and this particular molecule can be easily replaceable by increasing or by minor increase of the ionic strength. That is why the pink color peak is the first sharp peak that is coming out of the column based upon its charges. And second peak, you can see that the brown color one, which is coming next based upon the number of charges present on its surface. If you see the third peak, which is a blue color peak, that represents the blue color molecule which has 2 negative charges on its surface. So, compared to one negative charge, it has 2 negative charges. So, it needs more ionic strength to replace from the surface. That is why the blue color peak is coming as a third sharp peak. So, overall the right side picture represents the ion exchange chromatography chromatogram, or it is representing the chromatogram of one ion exchange chromatography experiment showing 3 different kinds of molecules, pure molecules that are eluted out of the ion exchange chromatography column. The last step in ion exchange chromatography is the regeneration. After eluting out of all the required molecules from the column, we have to regenerate the column so that we can use that column next time. For that, we have to use high ionic strength equilibration buffer to replace all the sample molecules from the column and re-establish the adsorption between the positively charged and negatively charged ions in case of anion exchanger, or in case of cation exchanger, it is the attraction between negatively charged ions present on the stationary phase and the positively charged ions present in the sample buffer or equilibration buffer. So, that completes the procedure of ion exchange chromatography. It is very simple. It has only 4 steps. The first step is the equilibration, the second step is the sample application, and third step is the washing and elution, and fourth step is simple regeneration. So, after regeneration, we can reuse the column for the next experiment. So far, we have discussed about the principle of ion exchange chromatography and the kinds of ion exchangers, and what are how to choose the ion exchanger, and also what is the pH of eluent, and what is the procedure of ion exchange chromatography. Let us discuss about the applications of ion exchange chromatography. The most important use of ion exchange chromatography in our daily routine life is deionization of water. Especially whenever we take water nowadays, we take RO water. This RO water is nothing but purified water from ion exchange chromatography. In this case, what happens is there are 2 kinds of exchanger in our RO system. That is called as, one is the cation exchanger, another is the anion exchanger. Either they are present in the separate column or they are present in the single column. The exchangeable ions on cation exchanger is H plus ions, and the exchangeable ions on the anion exchanger is OH minus ions. So, what happens is, whenever the water comes into the ion exchangers, for example, if it is a cation exchanger during water purification, say sodium ions are present in case of water, hard water, or calcium ions are present. These calcium ions replace the H plus ions from the cation exchanger. Suppose if there are the chloride ions present in our water, these chloride ions exchange the hydroxyl ions present in the anion exchanger. So, basically the sodium ions attached to the cation exchanger and chloride ions attached to the anion exchanger. Like that, other cations and anions, maybe the calcium cation or some other anions, which will be attached to the exchanger, and because of that the H plus ion and OH minus ion will be released, and these H plus ions and OH minus ions, they will combine together to give pure water. That is what we are drinking every day, pure water from the RO system. So, this is one of the common example where we can use ion exchange chromatography in daily routine life. The another example or use of ion exchange chromatography is in case of amino acid autoanalyzer. Amino acids, they will have charges on its surface at a specific pH. So, based on this principle, we can analyze the amino acids in case of amino acid autoanalyzer. And another important use of ion exchange chromatography is the separation of proteins. As we have discussed previously also, the proteins at certain pH values, they will have either anionic state or cationic state, depends upon its pKa value. And one more important application of anion exchange chromatography is the determination of nucleic acid base composition. It is very well known fact that the Chargaff rule, which tells that A binds with the T or G binds with the C, was mainly established based on the ion exchange chromatography. And in addition to the proteins and nucleic acids and also simple water purification, the ion exchange chromatography can be used for many other purposes. For example, the purification of other biomolecules like vitamins and biogenic amines, etc. This is all about, these are the main applications which we have discussed in this lecture, but there are many applications for ion exchange chromatography in several industries. Overall, in this lecture, we discussed about the basic concept and principle of ion exchange chromatography, types of ion exchangers, choice of ion exchangers, the procedure of ion exchange chromatography, and the applications of ion exchange chromatography. Dear students, I hope you enjoyed this lecture. For further study, please read Principles and Techniques of Biochemistry and Molecular Biology, edited by Keith Wilson and John Walker, the 6th edition, published by Cambridge University Press in 2005. In addition, we need to refer another book, that is the Physical Biochemistry Principles and Applications, edited by Sheehan D., 2nd edition, Wiley and Blackwell Publishers and West Sussex, UK in 2009.